Thank you very much for the introduction. Inflammatory responses in heart failure have been studied for many years, and I'm going to try to give you a story that's not complete, and hopefully we can come to the final chapters in the next few years. Progression of heart failure is listed in this slide, and it shows very simply here how a heart that's small becomes large after injury. And in this process of cardiac enlargement, many neurohormonal systems get activated, and it is along those lines that the inflammatory responses, we have identified and characterized many of those with the purposes of intervening, because we believe that these inflammatory responses may also provide a protective response initially, but in the long term, they are detrimental, just like what we have seen with the neurohormonal responses blocking the adrenergic system and the renin-angiotensin system. So inflammation is a very basic response. It's a response that occurs in a variety of conditions, as you can see in this portrait of Time magazine in 2004. Even in the late press, we were trying to understand the process of inflammation as a potential explanation for conditions that include heart attack, cancers, Alzheimer's. So the inflammation is a basic response to injury. And the question in the cardiac state is, how does this response contribute to initially a protective mechanism, but over time contributes to cardiac dysfunction? So in the field of cardiology, very few of us were interested in the inflammatory responses, frankly, because cardiology was about EKGs and then echo and then catheterization. But most cardiologists have been developed to train as technicians, and we were really not very interested in the biology of inflammation or the immune responses in cardiac conditions. But this is a seminal observation that's shown in here. This slide on the right is taken out of the Levine paper in the New England Journal of Medicine many years ago. This was co-authored by Dr. Milton Packer. And it depicts that in a large number of patients with heart failure, there was this hormone, tumor necrosis factor, that was elevated compared to patients that have chronic renal dysfunction or controls. It turns out that TNF was investigated in those patients not because it would cause cardiac injury, but because TNF was responsible also of the cachectic syndrome that was observed in animals that harbored tumors. So TNF induced cachexia, and therefore it was an attempt to explain the syndrome of cachexia in humans with cardiac condition. We took this further by investigating the levels of TNF in patients with heart failure, and we were able to define that as the illness progresses, as you can see here, there were higher levels of TNF in the periphery. And this should be reminiscent of what happens with the catecholamine levels in patients with advanced heart failure, that as the illness progresses, there is progressive increase in catecholamine levels. So this put forward the concept that TNF could potentially be involved in cardiomyopathy, and the observations are listed in here. TNF was increased in heart failure in the periphery and in the myocardium. We were able to investigate and define that in the cardiac tissue, there were high levels of TNF. TNF in experimental animals was capable of inducing left ventricular dysfunction. And in animals that overexpressed TNF in the myocardium in a cardiac restricted manner, they developed cardiomyopathy. And very simple initial clinical studies where we blocked TNF in the periphery led to some observations that suggested cardiac improvement. And that led to the conduction of two large randomized clinical studies, one with the use of infliximab, an antibody directed against TNF, and the other one with a recombinant molecule that's called etanercept. Both of these compounds are clinically available today for rheumatological conditions. These two compounds were developed in large multi-center national trials, and unfortunately, they did not show the blocking the biological effects of TNF in humans with heart failure led to an amelioration of the disease state. Not only that, the use of infliximab led to worsening heart failure, which now has led to a package insert modification of the specifications for infliximab that contraindicates its use in patients with heart failure. So highly selective inhibition of TNF, well, it appeared to be very, very consistent and important from a basic and early translational work, did not achieve clinical benefits in humans with heart failure. And then we reasoned that perhaps because we were very selective blocking TNF, that maybe we needed to take a more broad spectrum approach. And I will try to decipher to you this cartoon in a very simple manner. because the observation here led to a very large clinical trial and now leads to new observations that I will discuss with you towards the end of this presentation. So it turns out that cells that undergo apoptosis, they stimulate an immune response that's characterized in this cartoon that simply can be summarized by a production of T cells that decrease inflammation, where you change the immunoregulatory profile of T cells. But when a macrophage takes apoptotic bodies or apoptotic cells, it stimulates T cells to become anti-inflammatory. So one could take advantage of this by creating artificially apoptotic bodies, delivering to patients, and stimulate anti-inflammatory responses. So we did just that in patients with heart failure. And this slide is taken out of a paper that we published in JACC years ago, where we conducted a study at Methodist Hospital, at the Cleveland Clinic, at the Texas Heart Institute, and the findings are simply shown in here. This slide shows all-cause mortality, and in the active treatment group, we had 1 death and 7 deaths in the placebo group out of a 70-patient trial, randomized to this active treatment versus placebo. And the same observation can be seen in all-cause hospitalization. When we were confronted with these observations, we figured we have the panacea for heart failure. Now we have a broad-spectrum anti-inflammatory strategy that appears to be of great benefit in this small cohort of patients. We had to go and do a large multi-center study. I'd like to remind you that before we did this, in this particular experimental design, we did not conduct animal studies trying to see if this technique would ameliorate the condition in animals with heart failure. But because the observation was already in humans, we launched a very large study, what's called the ACCLAIM trial, that eventually was published in Lancet in 2008, but only to show one more failure of the anti-inflammatory strategy in heart failure. And this slide shows the primary endpoint of the study, where these two curves are completely superimposed. The red showing the IMT, or the immunomodulatory strategy, and then the placebo group. So this was quite frustrating, and the frustration was exemplified by a complete breakup of the company that led to that study. These studies are very expensive, and while we were observing small benefit in a population, we really were not able to conduct another large multi-center study. So we set back years into the process of understanding the inflammatory response to heart failure, and that led to this observation. And let me walk you through this slide, because I think it's an important finding that I think is relevant not only in chronic heart failure, but also in acute decompensated heart failure. This slide on the left is a slide from the ASCENT cohort study, and it shows the cumulative mortality over time of patients that come in with acute decompensated heart failure. And as you can see, you can separate those that come into the hospital that are troponin negative versus troponin positive. So heart failure patients with acute decompensator that have cardiac injury or are troponin positive have very high mortality over time. This is in-hospital mortality. So a very fundamental observation. Now, if I go back to one of the previous slides, and I remind you that cells that undergo death by apoptosis can stimulate immune responses in a different way. And necrotic cells also can lead to an inflammatory response. With this observation, we asked the question, what other cells within the immune system may be activating in the setting of acute decompensated heart failure? And we demonstrated, as you can see in the panel on your right, in a paper that was published a few years ago, that if we measure B cell and B cell activation, there were high levels of B cell activation markers that were present during the course of the acute heart failure state. These are individual patients. There's a group of 20 patients that we follow between the onset at baseline and during the course of the hospitalization. So we put the concept together that B cells may be activated in heart failure because they are stimulated by injury that's produced in the myocardium. And that led us to study and investigate whether or not cardiac tissue, end-stage failing hearts, contain markers of B cell activation. And specifically, we look for the presence of antibodies that may be deposited in the myocardium. This slide is out of a paper that we have in the European Heart Journal published a few years ago. On this panel, it's end-stage myocardium. Both were stained for an anti-IgG, developing green when it's positive. And you can see the normal heart does not contain IgG. And this is a representative slide that showed sarcolemma staining of IgG that's been deposited in end-stage failing myocardium. So we went on to demonstrate that in about 70% of the hearts we had IgG expression, predominantly IgG3, and some of those contain complement. This slide shows the dual deposition of IgG and complement. And this is of importance because when complement is fixated in the IgG, it potentially brings to you the idea that it could be of pathological significance. Complement fixation may destroy tissue and be a component of progressive tissue injury. So that paper has led to an active interest in ameliorating these B-cell responses in vivo with the purposes of obtaining a clinical benefit. Now, how do B-cells become activated? I'll walk you through this cartoon very simply. We say damaged cardiac tissue is picked up by macrophages that creates a set of inflammatory responses that may impact fibroblasts or cardiac tissue. And in support of that concept, there is a very recent paper that was published by Dr. Mann's group, published in SHA, that shows very simply the necrotic myocardial cells, when they are used to stimulate fibroblasts or cardiac inflammatory cells, they actively stimulate a very profound inflammatory response. And I really won't walk you through that slide, but I want you to just to look at these histological sections of necrotic myocardial cells injected into cardiac tissue, and you compare this profound inflammatory response to the saline inflammatory response. So necrotic cells are capable of stimulating an inflammatory response, and we think that that may be also an important element that activates B-cell function. So the really important question is, because the theory is very exciting today, we believe that cardiac tissue that's injured not only provides an aggressive cytokine stimulation, but we also think that it activates specifically B-cell responses. But the real question is, do we have evidence, or can we prove that IgG or B-cell activation is important in heart failure? And I'm going to show you very simple data in an animal model that demonstrate that experimentally that is in fact the case. In a laboratory, we can stimulate heart failure by giving angiotensin in mice. This is a normal mouse. This is a mouse that's ill, and this is how it looked by MRI, and this is a normal heart, and this is a heart that's obtained from this mouse that's very ill. This is also a paper that we have published in SHA. When you look at cardiac function as measured by MRI, animals that develop heart failure had LV dysfunction, and animals that do not contain B cells, as exemplified in this group, or that have a severe combined immunodeficiency, when they were induced to develop cardiomyopathy, they have preservation of LV function. You compare those to this one, reduction of EF in the cardiomyopathy model and preservation of LV function when you don't have intact B cells. We can measure B cells in activation B cell in the periphery of these animals. We can also measure intercardiac expression of IgG as shown in here in this very bright red, which is very consistent with what we saw in the human. And experimentally, we went ahead and treated humans with plasma exchange, a strategy that modulates B cell responses. And I'm going to show you brief data that supports the concept that in modulating B cell responses with plasmapheresis may cause some biological effect that's consistent with the role of B cells in humans with heart failure. This is change in ejection fraction from baseline to 3 months following plasma exchange. And this represents that we can reduce the concentration of intracardiac IgG in myocardial samples of those patients. And we're currently conducting a study with anti-B cell therapies in humans with heart failure to try to prove this concept. So let me summarize in the last few moments here that immune activation occurs in heart failure. There is no question that that's a prevalent problem. The triggers that may be related are perhaps responsible for the release of these necrotic cells that provide these damage-associated molecular patterns. We believe that anti-cytokine intervention was not successful because it's so highly selective. The system is redundant, and there are many other pathways. But we are currently investigating novel approaches to prevent B-cell activation in humans with heart failure. Thank you for your attention.
Thank you, Dr. Torre.